Partendo dal corpo, quindi la giornata sarà così suddivisa. Parlerà Komisaruk, che noi ringraziamo per essere qui con noi, perché lui si è dedicato allo studio della sessualità, e soprattutto non solo dal punto di vista biologico, come vi dicevo, ma anche dei circuiti e quindi della rappresentazione, ad esempio, del piacere e dell'orgasmo. Quindi Komisaruk, che è professore di psicologia dell'Università di Rutgers University, ma comunque lui è editor di molte riviste che si dedicano alla sessualità, a numerosi gruppi di studi, ma soprattutto noi lo abbiamo invitato perché è uno dei pochi studiosi al mondo che è andato lì a vedere, a cercare di capire cosa è rappresentato nel cervello durante l'atto sessuale, durante il piacere, durante la sollecitazione, la stimolazione corporea. Quindi oggi con lui vedremo questa parte, diciamo così, più biologica. Non importa cogliere esattamente quale sia il circuito o la molecola coinvolta, dico sempre, perché molti di voi magari non hanno mai studiato in dettaglio il sistema nervoso. Importa sapere che lì ci sta ad esempio un overlapping, si sovrappongono diverse aree. Cosa per noi è importante? Quali sono le aree coinvolte? Di modo che poi noi successivamente, in maniera più, direi quasi fenomenologica e anche clinica, entriamo nel merito della sessualità. Nel pomeriggio proseguiamo con un duetto in cui io parlerò stavolta del sessuale e della sessualità, cercando di mettere insieme la psicanalisi, che ha una lunga tradizione su questo. Se voi metteste insieme i volumi scritti sulla sessualità e sul sessuale in psicanalisi, io penso che riempiremo un bel po' di stanze. Quindi non potremo, ovviamente, io non sarò in grado di racchiuderle tutte, ma cercheremo di fare il raccordo con le neuroscienze e la neuropsicanalisi, di cui poi ci parlerà meglio Oliver Turnbull. A fine serata ringraziamo Bernard Chervet e la Rotrò e Katie Bogliatto e Blas, Heribert Blas, la coppia di presidenti e vicepresidenti candidati all'IPA, che sono qui in presenza con noi e alla fine della giornata, quindi alle 18, esporranno il loro pensiero su quanto andiamo dicendo ormai da anni sul rapporto tra psicanalisi e neuroscienze. Quindi siamo veramente contenti di poter sentire direttamente da loro il loro pensiero su questo. Domani tavola rotonda, caso clinico, diversi colleghi, quindi appartenenti a diverse discipline, anche a diversi modelli teorici, discuteremo insieme questo caso clinico e siete pregati, alla luce di quanto detto oggi. Per noi la domenica rappresenta la giornata di sintesi, alla luce di quanto detto oggi, di intervenire, chiedere, perché noi siamo qui per formulare domande, non per dare risposte. Quindi diciamolo subito, noi speriamo di uscire da questo con convegni arricchiti di domande, perché le risposte le avremo nel tempo, non siamo sempre chiamati a dare risposte subito, immediatamente, magari a cambiamenti di costumi che non capiamo, però a farci le giuste domande sì. Quindi, ripeto, in presenza, quando faremo la discussione, avrete la parola 3-4 minuti, mi raccomando, e chi è online scriva direttamente in chat, non serve usare il QA, può scrivere direttamente in chat, perché noi raccoglieremo le domande e le rivolgeremo ai relatori, perché in realtà introdurrà questo tema, perché è esperta in materia molto più di me, la dottoressa Claudia Spadazzi, che insieme a me, in realtà non mi sono presentata io nemmeno, ok? Dottoressa Rosa Spagnolo è fondatrice, insieme appunto a Claudia Spadazzi e a Francesco Castelletti Balarà, di questa avventura di cui siamo veramente felici, che è stata Italian Psychoanalytic Dialogues Association, perché ci permette di parlare tra di noi liberamente, affrontando temi spinosi a volte. E la dottoressa Spadazzi, che introduce il convegno, appunto, oltre ad essere full member della Società Psicoanalitica Italiana, nonché è anche sessuologa, quindi fa parte di diversi gruppi che si interessano di questo, è anche ginecologa, collabora con Berry, quindi diciamo l'ascoltiamo volentieri, perché anche se è un'introduzione al convegno, in realtà racchiude in sé tutto il senso di questo convegno, quindi l'abbiamo condensata in pochi minuti, ma ascoltate bene parola per parola, perché sarà tutto lo svolgimento del convegno racchiuso nella sua relazione. Do la parola quindi a Claudia Spadazzi, benvenuti.
Thank you, Rosa. I'm very pleased to be here today, and I will speak in English, even if my English is not so good. So in case you don't understand, please interrupt me, okay? And don't worry, I'm fine if you interrupt me if my English is not clear enough. So, yes, I'm very pleased because this topic of sexuality is really in my background, as I studied as a gynecologist first, and then I studied sexology, and then finally I did my training as a psychoanalyst. So I would like to say that on November 15, 2022, the world has reached 8 million human beings. And of these 8 billion, approximately 8 million were conceived after 1978. How sexuality, this vast, multifaceted, complex, and protean subject, is the cornerstone of the survival of any animal species. Commenting link between love, life, and pleasure, Andre Green underlines how, to each of us, the primary scene of our origin can be more or less disquieting. And any form of sexuality, however we wish to understand it, can only be understood through its biological foundation. Formulating his fundamental concept of drive in 1915, Freud highlights its biologic bedrock. Drive appears, you all know this quotation, drive appears as a concept on the frontier between the mental and the somatic as the physical representative of the stimuli originating from within the organism and reaching the mind as a measure of the demand made upon the mind for work in consequence of its connection with the body. Furthermore, as we know, he recognized the specific value of sexual drives, differentiated from the auto-conservative ones. The drive theory is currently undergoing revision, as we all know. For example, Mark Solm's revision, which was done in 2021, because of the rapid development of neuropsychoanalysis, which offers numerous elements to better understand these biological and mental foundations in their multiple declinations within the mind-brain-body unit of each individual. Just to make an example, the term psychosexuality, which is very largely used in the psychoanalytic community, for me, it sounds like a tautology, because there is no psychic sexual activity if there is not a psychic activity. So we cannot split anymore the mind from the body and the brain, and it is a whole unit. And this is very important, even if we think to the Freudian concept of the drive and what happens in during any sexual activity in each aspect of it of any event. So in the last 70 years, we have witnessed a paradigm shift from what the slide? No, he said to. Yeah. Okay. You have to. Okay. All right. So until the 50s, we have this equation: sexuality equal procreation. And so this was the situation since the origin of the mankind until the 50s. Then what happened? There was the onset of. Which one?
The lower.
Okay. Okay.
The lower.
Okay. So yeah, we had after the 50s sexuality without procreation. So there was the advent of contraception and women's liberation, because it was an enormous change, the possibility to have sexual relationship without sexual intercourse, without the risk of a pregnancy. And then, again, there was another shift, which is procreation without sexuality. So we had the assisted procreation techniques, and so we had homoparental families and single parents and everything which came out. So all this change which happened in a very short time, so we are speaking of just from the 50s to the beginning of the new millennium, there was an enormous change, and this has an enormous impact on the unconscious of people, because it was a very short period of time and an enormous change. So at about the same time, we witnessed not only the manifestation of form of sexuality and behaviors well detached from reproduction, but also to the emergence of forms of sexuality and behaviors totally new. More and more numerous issues need reflection on the biological and cultural level, on the subject of identity and on the object choice. So, in other words, on the sex and on the gender. Homosexualities, transgenderism, gender fluid, transvestism, gender dysphoria, intersexuality, and I'm here referring to a more biological aspect, which is the real hermaphroditism and the pseudo-hermaphroditism, which are specific biological conditions. All these challenge fundamental concept of psychoanalysis and force a continuous revision and rethinking from a mental functioning point of view, both theoretical and clinical. And this rethinking does not only concern the psychodynamic foundation, which with difficulty faces the need of updating, but also the neuroscientific definition of the multiple and complex aspect of this topic. The dichotomist view of male-female sexual difference, as old as human history, found scientific grounding in the discovery of the XX and XY chromosomes in 1905. The discovery of the heterochromosomes, or sex chromosomes, is due to a woman, Nettie Stevens. And I want to show you her, because she was a very clever woman, and she made this very important discovery, but the Nobel Prize went to another guy, as it's usually often happened at the time. And it is perhaps no coincidence that in the same year, Sigmund Freud published 3 essays on sexuality, which carried so much weight in the development of the psychodynamic theory. Brain differences between the two sexes have been studied for over a century, mainly as anatomic or neurocognitive point of view, and they are still a topic of research and debate. The historical male-female dichotomy has over time been overcome beyond the obvious difference in the genitals. In fact, the concept of a continuity of the two genders, such as maleness and femaleness, a spectrum, has gradually grown. This continuity accounts for fluctuation due not only to the cultural environment, so on the gender aspect, but also to environmental elements that may influence the biological sex aspect. For example, pollutants. We know that pollutants have some influence on the sexuality. And also food with a hormonal effect. If you think to chicken fed with estrogens, for example, or veals fed with estrogens. And also enzyme deficits, for example, the 21 hydroxylases. So there are so many conditions which can influence the biological sex. The sexual definition of the individual can be understood as positioning on a spectrum between maleness and femaleness. And Freud himself refers to a psychic bisexuality, as we know, founded on a significant biological basis. Even more recent is the concept of brain mosaic, which is basically due to Daphne Joel, Joel. According to this researcher, a new conceptualization of the sex differences is needed in order to overcome the two classes, male brain and behavior and female brain and behavior. So the traditional dimorphic view of the brain should be substituted by the enormous variability of the human brain mosaic. Although there are, and this is Daphne Joel writing, this is a quotation, although there are sex-gender differences in brain and behavior, humans and human brains are comprised of unique mosaics features. Some more common in females compared with males, some more common in males compared to females, and some common in both females and males. According to Joel, regardless of the cause of observed sex-gender differences in brain and behavior, so let's say nature and nurture, human brains cannot be categorized into these two distinct classes, male brain, female brain. Sex differences are currently investigated through several methods: research on anatomy, connectome, neurochemistry, hormonal effects, and neurocognitive functions. For example, numerous differences have been highlighted in the hippocampus and long-term memories, but this needs some more research. It is still open to clarification. Excuse me? Nowadays, there is a tendency to consider the brain as an endocrine organ. Hormones have a paramount function in the primary emotional systems, as conceptualized by Panksepp. The main system is at play are the seeking system and the lust system. The role of the seeking system in the search for the right partner, and it is basically dopamine dependent, and this is dopamine that drives the individual in this direction. Numerous studies agree that subjects with depressive aspects in which the function of dopamine is reduced, they don't involve themselves in the search for partners and show a hypoactive sexual desire, while in manic states, this search is increased to the point that sometimes they lead to inappropriate behavior. The last contributes distinctly to female and male sexuality and associated erotic feelings. Oxytocin is the main hormone involved, a neuropeptide which is produced mainly from the preoptic area in the male and from the ventromedial nuclei in the female. Oxytocin is then released in the bloodstream by the neurohypophysis and is released with a peak during and after orgasm. And we are very lucky today because we have Oliver Turnbull in the afternoon who will speak very specifically about this part, and just later Barry Komisaruk who will speak about the brain representation and organs. So we're really lucky today to share with them their skills. So the balance between seeking and lust is related to Berridge's wanting-liking theory, where seeking can be linked to wanting, and so the push to search for, and lust is linked to liking, so the experience of pleasure. Berridge's theory is well known for its connection to addictions, but the psychic mechanisms involved are similar, so wanting liking drugs or wanting liking sex. As clinical experience shows, if the affective systems are driven toward substances, desire and sexual interest are reduced. And here, I'm not referring only to alcohol or to substances, but also to the addiction sine substantia, so addiction, for example, to pornography and gambling and all this internet addiction and so on. So I don't intend here to go into the subject of perversion, to put it psychoanalytically, or paraphilias, to put it with the DSM, a subject too vast for this introduction. But let us focus on sexology and on sexual dysfunction, which are not having per se a psychopathological status, but they can create enormous sufferance, and they can lead to depression, to anxiety, sometimes even to suicide. The history of clinical sexology started in the US just after World War II. And so it is a discipline of American origin which developed between the 50s and the 80s. See. So now I don't have the time to recall the whole history of sexology, but just mention that it started with Alfred Kinsey, a biologist. Then it went through Master and Johnson in the 60s. Then there was Helen Kaplan, who was a sexologist, but then a psychologist and then a psychiatrist. She became professor of psychiatry. And she had a psychodynamic approach. And now, still now, her book, New Sexual Therapy, is the basic textbook for sexology. And it's very important, her contribution about the psychodynamic approach. And then finally, in the 70s, there was George Engel, who conceptualized the biopsychosocial theory, which is now very much in use, especially in the States, especially about sexology. So, so here you see just a table about the request about sexology in male and female. And now this is the criteria of morbidity. So a symptom has to persist for a minimum duration of 6 months. It must be experienced in almost or all sexual encounters, and have caused the individual clinically significant distress. These are the main, for those who are not sexologists here, the main dysfunction. So for female orgasmic dysfunction, female sexual arousal interest or arousal disorders, and then the genitopelvic pain or penetration disorder. This was a change in the last DSM, a change which I don't agree very much, but we will have the opportunity to speak about a problem like this tomorrow in the round table with the clinical case. And then we have the male dysfunction, premature ejaculation, erectile disorder, and delayed ejaculation. This is the contemporary basis of sexology. And as we know, in most psychoanalytic training, it's not a topic that is addressed. As Mark Bleckner states, most psychoanalysts know relatively little about sex. And he means not only about dysfunctions, but also about this new form of sexuality and sexual behaviors which were once classified as perversions. So psychoanalysis, as already pointed out by Kaplan in the thesis, offers valuable tools for the understanding and treatment of sexual disorders. Because there are some determinants which are unconscious, of course, and which require, on the one hand, specific training, but on the other hand, the analyst confidence with the analyst counter-transference. And sometimes patients need an integration of different skills or a teamwork in order to consider the numerous aspects of sexual dysfunctions to find a better specific treatment. So I want to conclude my introduction with a question that I address to our speakers first of today and of the afternoon. and to all you participants, and also to the candidates for the presidency and to the vice presidency of the IPA that we have today the honor to host with us today. And so the question is, has the time come for a psychoanalytic, or even better, a neuropsychoanalytic sexology? Thank you for your attention.
Io direi di dare subito la parola a Barry Komisaruk, che ho già presentato, introdotto, e che siamo veramente felici di avere con noi per questo punto di vista fra il corpo e la mente, che lui è riuscito a studiare molto bene. È stato un piacere averlo con noi ieri sera e parlare di questo con lui, e speriamo in un qualche modo che anche lui possa arricchirsi di ciò che poi noi andremo dicendo, perché senz'altro io mi sono arricchita moltissimo leggendo le sue slide. A te la parola.
Yeah. Thank you, Thank you, Rosa. Thank you, Claudia, for your kind invitation to this conference. I'm really looking forward to it. And also thank you, Rosa and Francesco, for your very wonderful, and all of you for your wonderful hospitality. I really appreciate it. And I'm looking forward to hearing the concepts of neuropsychoanalysis. It's really a very intriguing idea to me, and I'm looking forward to hearing what you all have to say. So I would like to start by telling you how I got into this area that I'm going to talk about. When I was starting out on my doctoral dissertation, I was intrigued by two recent studies showing that crystals of estrogen implanted into the brain of a cat or a rat would stimulate their sexual behavior. And this was very intriguing to me. And so this is what I did for my doctoral dissertation, implanting progesterone crystals into the brain of ringdoves and inducing their reproductive behavior. And I was convinced that it really does happen. So then I wanted to know, what does the hormone do to the neurons in the change their behavior? And the only person doing it at UCLA, and he was studying a neuroendocrine reflex and the relationship between brain activity and hormone secretion, how the brain controls hormone secretion, say in the case of reflex ovulation, where cats only ovulate if they mate, and then also how the hormones act back on the brain. And he was studying progesterone in context of pseudo pregnancy reflex, in which mating stimulates the hormones of pregnancy, and that involves vaginal stimulation, activating the brain to release hormones that the gonadotropins that stimulate the ovaries to secrete progesterone. And so we were studying that reflex in anesthetized rats. And I continued doing that when I returned to my position at Rutgers University, where I was setting up my lab. And I continued. This was in anesthetized rat. Then I tried it in awake rats, and I found a very interesting phenomenon. If I can get that started. Let's see, how do I? There. Well, I just pinch the foot, get a normal withdrawal reflex, and then I applied the vaginal stimulation that we were using for the neuroendocrine reflex, and mild stimulation. To my surprise, the rats became immobilized, and then when I tried to pinch the foot again, the response was completely blocked, and no vocalization, no withdrawal. So I said, what's going on here? Is it blocking pain, or is it inability to move? And so I did recording in the sensory thalamus of single neurons. They respond to both touch and to pain. This was brushing the fur. Here I applied the vaginal stimulation in the middle, and it didn't have any effect on the touch response. But when I applied, this was scratching the cornea in anesthetized rats, the vaginal stimulation blocked the pain response, but not the response to scratching the cornea, but not to touch. And I said, that's evidence that it's inhibiting pain and not just producing anesthesia and not simply immobilizing the animal. So I said, the only way I'm going to know scientifically whether this is really blocking pain is by asking women. And that's how I got into human studies. And with Beverly Whipple, who did her doctorate with me, this was her project for her doctoral dissertation, I devised a vaginal stimulator that we could measure the force with a transducer in the neck, and they would hold this and press it against the anterior vaginal wall. And then we measured the pain threshold by this device that increased the force on the finger. There's a small point here, 1 millimeter point, and this is the weight moves out along the arm and increases the force. We calibrate that. So we and ask them to tell us when it first hurts, and then we lift the arm and try it again. So that's how we got the pain threshold. And what we found is that when the women applied the just pressure to the vaginal wall, the anterior vaginal wall, we got about a 50% increase in threshold. When we said do it in a way that feels pleasurable, it went up to a stronger inhibition. And then when we said, and 4 of those 10 women went to orgasm, and we got an over 100% increase in pain threshold. So that was convincing evidence that vaginal self-stimulation produces pain blockage, produces analgesia. So I was interested in what nerve is carrying that pain-blocking sensation. And so the pudendal nerve carries sensation from the clitoris, and the pelvic nerve carries sensation from the vagina and the cervix, and the hypogastric nerve carries sensation from the cervix and the uterus. So in order to see what the difference in the function on which ones are carrying the pain-blocking signal, I studied women with a completely severed spinal cord, and at 2 different levels, to block the pudendal and pelvic nerve input at this level, and then block all the input at the highest level, the higher thoracic level. That would block all the access of all the genital sensory nerves to the brain. And we measured the pain threshold then. And to my great surprise, the strongest pain blockage occurred in the women who had the completely severed spinal cord. How could this be? All the pain pathways through the spinal cord are cut. In addition, those women, they all said that they have menstrual cramps. And this was very puzzling to me, because how come they or their doctors never realized that they have sensation in the genital region, although they didn't have any clitoral sensation or any other sensation below the level of injury, but they did have, of all things, they said, the only thing that I have left is menstrual cramps. And they all had they could all feel the stimulation. They could all, and 3 of the 5 women with this completely severed spinal cord said that they had orgasms from the stimulation. So I decided the only possible pathway was via the vagus nerves, which are the cranial nerves that go outside the spinal cord. So the only way to know that was by by doing functional brain imaging to look at the activity of the sensory projection zone of the vagus nerve, which is the solitary nucleus in the medulla of the brain, of the brainstem. And the question was, does the vaginal self-stimulation activate the solitary nucleus? And we found that in all 5 women, it did indeed activate the solitary nucleus. So that was evidence that the vagus nerve does, in fact, carry sensation from the vagina and cervix. So, but at the same time, this was the first evidence of where orgasm occurred in the brain, because he had the functional MRI recordings, and we we saw activation of the brain related to orgasm. So I got interested in orgasm and where this is happening. That led us to study the sensory pathways to the brain from the genitals. And here's a sensory cortex. And this is Penfield's homuncular map, where he, in Awake Humans, he was a neurosurgeon at McGill and doing epilepsy surgery, removing a part of the brain to control the epilepsy. So before he did that, he electrically stimulated with a handheld electrode different parts of the sensory cortex. And if he stimulated the lateral part, the people said that feels like you're touching my face. Here, they're touching my hand. And he even done in the midline, this region, it was almost all men that he studied said that they're touching the genitals. We tried to see if this was true in the functional MRI also. It's the paracentral lobule that he was measuring the activity, and right here is where he got the response. So when we did the functional MRI, in men, we got exactly the same location, an activation in the same exact region that Penfield had on his map, confirming that. And when we studied in women, we found that there was clitoral, vaginal, or cervical self-stimulation all activated the paracentral lobule, the same area as in men, although slightly different regions. This activity was the hand activity because they're applying left hand or right hand. They're applying the self-stimulation. So this is a blow-up of that same region, showing the response from the clitoris, vagina, and cervix. And I related this to the women's descriptions of orgasms elicited by stimulation of the clitoris. And they said it's a localized sensation, and the pudendal nerve that carries this input is a somatic nerve. And then if they did vaginal self-stimulation, they said it's more deep and heaving internal. It's a different quality of orgasm, and that's mainly the pelvic nerve. And if they combined them, they described having mixed orgasms. And when they did cervical self-stimulation, so the women described the orgasms as in these much more abstract terms, like shower of stars, or seeing a light expanding in my brain, or images of universal spaciousness, much more abstract descriptions, much less concrete than localized externally. And when we stimulated all, when they combined all for the orgasm, they said they're much more complex, intense, and interesting. So I think a way of accounting for that is that each of these individual regions represents the activation of neurons in those regions, and if they're each capable of eliciting orgasm, when they're all stimulated together, it's a more complex, more intense, and actually more pleasurable orgasm. We had different controls. When we do controls, we're always surprised with what happens with the controls. We asked, as part of the mapping of the body, they did finger stimulation, toe stimulation. We did toe stimulation. We asked them to do nipple cell stimulation. I expected that we'd get activation of the thorax. But to my surprise, we got activation of the genital region. And here is just a few of the women that we did get. The white is the activation by the nipple. We did get activation in the thoracic region up here, but in these cases we got the activation by the nipple was congruent with the activation of the clitoris, vagina, or cervix, and this was really a big surprise. And when I tell my my male neuroscience neuroscience colleagues, they say, well, that's, we have to change our concept of the Penfield map. And when I tell my female neuroscience colleagues, they say, yeah. So this may be a way of accounting for why nipple self-stimulation is pleasurable. And some women can have orgasms from it. We also did mapping of the male genital sensory cortex, and we get exactly, depending on the regions, they're all slightly different, like as in the women, but they're all in the paracentral lobule, same region. So it's the same kind of response, different responses with different sensory nerves, and each responsive region is slightly different, but all in the same general region. We also asked the men to apply nipple self-stimulation. We got the same thing as in women, that the nipple self-stimulation in men also activated the same region as the genitals. And some men describe nipple stimulation as pleasurable and erotic. The typical view of the sensory innervation of the penis is the pudendal nerve that carries and it's true that it carries sensation from the skin of the penis. But it's also true that the pelvic nerve, which produces the erection, innervating the corpora cavernosa, but that's the efferent. And nobody really pays attention to the afferent aspect of the pelvic nerve. But we see that there really is a difference in the input from the pudendal and the pelvic nerves. The evidence is that, well, first of all, in Netter's diagram, he shows that there is a sensory branch of the pelvic nerve. Not only the parasympathetic innervation of the corpora cavernosa that produces the erection, but there's also the sensory branch. And so what we did was, it may be a little hard to see. Thank you. Okay. We asked the men, in order to tell if there's something specific about input from the corpora cavernosa, to give maybe there's a different erotic quality to the deep penile stimulation as opposed to just the skin. So we had the men squeeze the penis, the shaft, and compare that to squeezing the glands, because the only difference between the glands and the shaft is the presence of the corpora cavernosa, which is the erectile tissue. And when we did that, when we had them do that, and we electronically subtracted the activation of the glands from the activation of the corpora, of the shaft, we got a difference in the location of the activation. You can see the different level of the brain and also different location. In other words, there are two different inputs from the penis, not simply the skin, but also evidently the pelvic nerve as well. And the pelvic nerve is what carries sensation from the vagina, and also probably the corpora cavernosa in the female, in the clitoris. So it's likely that these two nerves are the afferent nerves from the penis and the clitoris. We also asked the men to do two types of penile stimulation. One that just feels quotidian, about prosaic stimulation, like just clothing or sitting down, and do it in a way that feels erotic. And when we compared that, when we subtracted the prosaic from when they said it's erotic, we got two regions. The nucleus accumbens was activated, and the medial prefrontal cortex was activated. And this is the regions of the brain that apparently are activated when the stimulation feels erotic. And that's part of the pleasure, so-called pleasure regions of the brain. Now, the idea of the somatosensory cortex is that it's somatosensory. It responds to tactile stimulation of the body. But again, we did a control of don't do the stimulation, just think about the stimulation. And to my surprise, again, when this was in women, when they thought about clitoral stimulation or vaginal stimulation or nipple stimulation, we got activation of the paracentral lobe, the same place as when they actually did the physical stimulation. And we said, there's got to be some kind of difference. So here here's the response to physical self-stimulation, clitoris or nipple. And then we also got activation when they thought about the stimulation, but there was a big difference. In the physical self-stimulation, there was relatively little activation of the prefrontal cortex. But look at during the imagination, there was tremendous activation compared to when they actually did the stimulation. So this is the so-called executive part of the brain. So this is presumably at least one of the ways in which we can tell the difference between where something is actually happening as opposed to when we're thinking about it. So what are the brain regions that we found are activated in women? We really continued this in a more comprehensive way in awake, intact, able-bodied women. So I have this using a so-called hot metal analog, where as if you're heating up metal. This is a visual way of representing brain activity. That the dark red is the coolest, and as you start heating it up, it gets brighter red, and then orange, and then yellow, and then white hot at the highest level of activation. So we did this analysis, and this is leading up to orgasm. You can see there's a feedback.
Yes, there is a feedback.
Somebody. Leading up to orgasm, the brain is getting more and more active gradually. And here at orgasm, this is during orgasm, it's relatively most highly activated, and then after orgasm, it cools down. I know it's a little too bright, so you can't see the slide so well, but this is how I really analyze the data, dividing it into 80 columns, the so-called Brodmann areas. There are about 80 divisions of the brain based on morphology. So each column is the activation of one of the Brodmann regions. There are about 80 columns, and this is the left side of the brain, the right side of the brain, and then each row is the functional MRI can take scans every 2 seconds, scans through the whole brain every 2 seconds, one after the other. So each row is a 2-second scan, and this is a period of 5 minutes. And so this is showing what happens this is one woman, as she approaches orgasm. You can see different parts of the brain become activated at a different sequence. But here is orgasm. And there's most activity at orgasm throughout the brain, and then it starts cooling down after the orgasm. And this is a summary, same kind of summary where I lined up the orgasms here. The green is when they're doing the self-stimulation. The red is when they press the button to say when the duration of their orgasm, when the orgasm starts, and the duration. So you can see that there's a maximal activity at orgasm in these. This is 6 different women, and here. This is a summary of in 11 women their orgasm. We asked them to press the button when their orgasm starts. And it's linked to the computer timer. So then we could get so we could see the 10 seconds immediately before orgasm and the 10 seconds immediately after the start of the orgasms in the group of women. And you can see there's this general, these are different regions of the brain listed here. But you can see there's a general increase in activity at the point at the initiation of orgasm. So we said, if we subtract this activity of high arousal immediately before from the activity immediately after, we could see what's the activity that's more selective, more reflective of the orgasm per se, subtracting out the arousal. And when we did that, so that's what we did. We got the differential regions, and this is some of the different regions. Now, I want to see how could, here you're getting activation of different parts of the brain. How are these related to the components of orgasm? So here are some of the components of orgasm: tachycardia, hypertension, analgesia, pupil dilatation, oxytocin release, dopamine release, erotic pleasure, orgasmic seizures. So looking at specific some of these brain regions to try to account for some of these phenomena, we see that the region of the anterior hypothalamus. Here's a close-up of that. The region of the anterior, this is the region of the paraventricular nuclei. This is the anterior commissure. And here's a schematic diagram from Netter showing these are the neurons, the paraventricular neurons that produce and release oxytocin into the posterior pituitary, as Claudia was saying. And then this is during parturition, during childbirth, when the fetus's head is pressing against the cervix. That stimulates a reflex that releases oxytocin into the blood, that stimulates uterine contractions, that forces the fetus out, and that activates a rare positive feedback mechanism where the stronger the stimulation is, it releases more oxytocin, and then that stimulates uterine contraction still stronger, that activates it more, and so that's a positive feedback in the case of the childbirth release of oxytocin. That's called the Ferguson reflex. At the same time, the oxytocin stimulates the myoepithelial cells surrounding the milk glands that squeezes the blood, the milk into the baby's mouth. So it's a suckling reflex as well. But there are also some of these neurons bifurcate and send activation down the spinal cord. So oxytocin is also released as a neurotransmitter as well as a neurohormone. And I think this is very interesting because we said, well, what's the function of the oxytocin released into the spinal cord? Here's first of all, the oxytocin is, since this is stimulation of the birth canal, the cervix and vagina, at orgasm, Carmichael and Davidson and others showed that there's a peak of oxytocin at orgasm in women, and while oxytocin increases in men at orgasm, it doesn't peak in the same pattern. So these neurons become activated at orgasm, and we see that in the functional MRI. But we also get this, we've seen that in response to vaginal stimulation, we get pupil dilatation. And we also found in women that their pupils are dilated at orgasm. They become greatly dilated. So in the case of the rat studies, vaginal stimulation produces the dilatation, and we tried to see, well, maybe is that a possible function of oxytocin in the spinal cord? So this is the response when we do vaginal stimulation. We get this immediate response of the pupil. The pupils dilate immediately upon the vaginal stimulation. The oxytocin, the control of the pupil is from the thoracic level of the spinal cord, thoracic 1 to 3. That's the preganglionics are here. So we wanted to see what happens if we inject oxytocin there, And when we injected the oxytocin, we looked at all kinds of other possible responses to oxytocin. But we found that when we injected the oxytocin, we got an immediate pupil dilation in the rats, injecting the oxytocin into the spinal cord right here. And it was a dose response. A higher dose gave a stronger response. So we said, could this possibly, to confirm this, what we did was inject an oxytocin receptor blocker here, applied the vaginal stimulation, and we got a significant attenuation of the pupil dilation to the vaginal stimulation, which means that the vaginal stimulation releases oxytocin not only into the blood, but it releases it into the spinal cord, and it stimulates so oxytocin is a neurotransmitter to stimulate the sympathetic. It's a sympathetic stimulus because this is the sympathetic preganglions that produce the pupil dilation. But it's interesting that why do the pupils dilate during orgasm and during, this would imply that it's also released in the spinal cord during nursing. It may be that the pupil dilation is a way of facilitating eye contact during intercourse and during nursing. Just a speculation. There's no really way to test that very carefully. If you have any suggestions, I'd be happy to try it out. Also get the tachycardia. We get activation of the posterior hypothalamus and the hypertension. We know that dopamine is released during orgasm, at least during sexual stimulation, at least in animals, and drugs that facilitate dopamine release in humans facilitate sexual response and orgasm, and drugs that block the dopamine receptors inhibit sexual response and orgasm. So the pathway for the dopamine system is from the ventral tegmentum in the septum, in the midbrain, to the nucleus accumbens in the forebrain. And we found that ventral tegmental activation occurred during orgasm. This is the activity, the selective activation during orgasm. We get ventral tegmental activation. And we also get activation of the. We also get. We also get activation of the nucleus accumbens, and here's a blow-up of that. So you can see the activation of the nucleus accumbens. So that can account for the increase in dopamine at orgasm. There are also orgasmic seizures. That's been related to hippocampal activation and also amygdala activation that we see. But there's also evidence of the insula being involved in orgasmic seizures. This is a study by Anzalotti recently. This is a focus of epilepsy in the insula, and this woman had orgasmic seizures. She felt that her seizures were very erotic, and they measured the brain activity, and in these they found activation in these different cortical regions indicated in blue. Those are exactly the same regions that we see activated under normal condition, under normal orgasm. So these are some of the cortical regions activated during orgasm. But of course, you remember we got the pain blockage, the analgesia, during highest during orgasm. The periaqueductal gray is a very interesting region. That's of the midbrain. It's because it seems paradoxical, but it's really a homeostatic mechanism. It responds to pain. Periaqueductal gray neurons respond to pain, but they also stimulation there inhibits pain. So it's a key it's like a thermostat. It's a key to respond to and inhibit pain. We get activation there. We also get activation in the dorsal raphe. And in the classic descending pain mechanism by Bassbaum and Fields, they activated the activation is the pain gate mechanism is that the periaqueductal gray is activated. It sends stimulates the raphe neurons that contain serotonin that descend into the spinal cord. And here is a close-up. The descending serotonin, and this is also norepinephrine, activates opiate interneurons that block the incoming pain signal and block the activation of the incoming intense stimulation to activate the spinothalamic neurons that convey pain to the brain. So we see that this system is activated by vaginal stimulation, and we see it activated during orgasm. So in addition to that, we see activation of the anterior cingulate cortex and the insula at orgasm. And this is really interesting because here is the classical pain projection site. We see almost all the regions of the classical pain sensitive part of the brain are also activated during orgasm. So what is this intriguing connection between pain and orgasm, or pain and sexual response? There's a very important study by Berwick and Light in 1993 where they found that orgasm and pain use the same spinothalamic sensory pathway. That they studied 90 patients with spinal cord injury, partial spinal cord injury. 5 of them had injury that just of the front half of the spinal cord, where the spinothalamic pathway is in the anterolateral region. This is the spinothalamic pathway. The anterolateral region of the spinal cord, and the touch pathway is in the posterior. These are posterior columns. They carry the tactile sensation. So they had, in these 5 patients, 3 male, 2 female, they all had severe or complete absence of small fiber. They all had a completely severed anterior part of the spinal cord, but preserved the large fiber. This system was preserved. This is a schematic view of the tactile pathway, in just showing half of the spinal cord. This is the tactile pathway is intact, while the pain pathway is blocked. This is if this anterior part of you can get selective blockage of the anterior of the spinothalamic pathway. These 5 people lacked pain below the injury level, but they all had absence of orgasm. And then they asked the people to do intense stimulation of the genitals with a vibrator, and they said they could feel certainly they could feel the stimulation of the genitals, but they couldn't have an orgasm anymore. These are all people who had orgasms prior to their spinal cord injury. So this is really interesting, something unique about the ability to have orgasm and sharing it with the pain pathway. These individuals are all just having orgasms. So what it made me think about was as we see the increase in orgasm and the increase in pain blockage, there's got to be an inhibitory system that's being activated along with the excitation to produce the orgasm. And in men, when we look at the time course of orgasm, we see that here is at orgasm, a sudden increase at the beginning, when they press the button, there's an increase in overall brain activity at orgasm, and during orgasm, the activation goes way down. During the orgasm, they're continuing the stimulation here. They're getting decreasing activation. It must be increasing inhibition going on during the orgasm, and then after the orgasm, this is the refractory period. This is the inherent connection between excitation and inhibition. And this made me think about inhibition and excitation. And I have to realize that, well, I began to realize that at all levels of activity, inhibition is going on. That it prevents spasticity, just in general motor activity. As just picking up a chess piece is very low level of motor activation, and there's also inhibition going on to prevent spasticity for the very precise movement. But then taking a walk, the graceful movement, the higher level of excitation, but there's also have to be a higher level of inhibition. And running a race or running upstairs is a much higher level of motor activation, but there also has to be increasing inhibition. So this made me think of a schematic representation of orgasm as an increasing intensity of stimulation, increasing intensity of inhibition. Here's my hypothetical formulation is that since they're both increasing together, at a point, perhaps at a point where the excitation supersedes the inhibition, that's where pain is perceived. But in a normal orgasm, say at the point of orgasm in men, maybe the excitation just peaks out over the top, and that is what triggers the very high level threshold of the sympathetic activation for ejaculation, because it's necessary to have the activation of a high threshold sympathetic mechanism to produce the ejaculation. And then the inhibition occurs afterwards, producing the refractory period. And perhaps in women, the same pattern occurs, but the inhibition after the orgasm is not as strong, and that enables, with continued stimulation, enables women to have additional orgasms that may actually recruit more and more activation, and the subsequent orgasms may actually be stronger. So that's my hypothetical formulation, and I use an analogy of hot and cold water faucets, where you could have, because you have different intensities of pain, a mosquito bite as opposed to toothache, that it's the same pattern of the excitation succeeding or superseding the inhibition, but at different intensities. But always the same pattern. And perhaps the same thing is true in the case of orgasm. And I'm thinking of a pathological condition of anhedonia, which is, we had collaborating with Erwin Goldstein at San Diego, and he has patients with orgasmic anhedonia, which they say they have orgasms, but one woman described it, I expected a mountain and I got an anthill. So perhaps here's a typical orgasm, and an anhedonic orgasm is an orgasm, but it's just at a very low level, very weak intensity. So you get a little peak of excitation over the inhibition at orgasm, but it happens at a very low level, just like perhaps premature ejaculation. Same kind of thing. It's the ejaculation occurs, but it occurs at too low level of inhibition. And actually, injecting antidepressants can delay the orgasm, and it becomes stronger. So it's a way of treating premature ejaculation. This is just an interesting aside that there are also non-genital orgasms. There are many many descriptions of non-genital orgasms in systems of the body other than the genitals. We published a paper on that. This is particularly interesting. In men and women with spinal cord injury, many men and women with spinal cord injury have told me this, that if they get if the region around the skin region around the level of the injury is extremely hypersensitive, that if it gets accidentally brushed by clothing or even a wind gust blowing the clothing against that region, it's excruciatingly painful. But if the right person stimulates it in the right way, then it feels orgasmic. So so maybe it's the difference between the pain and the orgasmic pleasure is the relative intensity of the inhibition, and this could be obviously cognitively applied. Basically, all regions of the body have been shown to elicit orgasm, are elicitable. I mean, orgasm is elicitable, but by essentially all parts of the body. And these are other types of orgasm. But I think there seems to be a unifying concept here to me, that they all involve tension of the body, muscle tension, and the proprioception, the feeling of the muscle tension. And it can happen in any place of the body, any place where there's muscle activity. Maybe this is the unique feature of orgasm. And in the case of muscle tension, this is the Golgi tendon organ reflex. That it's the Golgi tendon organ is in the tendons of the muscle, and when it has a very high threshold. When it reaches a high threshold, it triggers the activation of the Golgi tendon, the Golgi neurons in the spinal cord, and that activates an inhibitory interneuron that turns the muscle contraction off. So, and that is kind of the paradigm of orgasm, that you get to a high level of excitation and it ends up in an overall inhibition. So, and this is a protective reflex. It prevents ripping muscle tendons out of the muscles. Or that is, ripping the muscles out of the tendons or the tendons out of the bones. So it's a protective reflex. So maybe the orgasmic process is a general sense, and the genital stimulation or produced orgasm is a special case of the more general orgasmic process of gradual building up of excitation leading to a peak, a climax, and then a resolution that can occur throughout the body. It's a general principle, and it may all be based on the simple spinal reflex just extended to the brain, getting more and more complex. Now, I got into, I won't describe this briefly, and just kind of the concluding part, I won't explain how I got into studying genital pathology to a great extent, but there are different kinds of genital pathologies. I got involved in persistent genital arousal disorder, and or PGAD, that occurs in men and women. This is genital sensation that is continuous, and it can actually produce multiple orgasms, spontaneous orgasms during the day in men and women, and it's stimulation that feels very bothersome. It's just continuous, like, sweetness. It becomes intolerant after a while if it's just incessant, and it can last for days, weeks, months, years, and it can be extremely distressing. And we know of 12 women in recent years who have committed suicide from it. They can't get any relief from this persistent arousal and persistent genital activation. And then there are also, and some of them have even ejaculations in the case of men. Plus, there's also conditions of the genital pain and loss of sensation. I think there's a syndrome here. One of the puzzling things is that has been puzzling, that I think we have at least a partial answer to, is that there are women who complain of this continuous problem of this continuous clitoral stimulation, and it's either continuous and very distressing or painful. And the problem is that the typical treatment is either giving local anesthetic, doesn't help, or giving or anesthetizing the pudendal nerve, which also doesn't help. It makes the clitoris numb. They can't feel touch, but they say, I still have the pain there. And there's even a case in the literature where the woman was so desperate that she insisted that the surgeon remove her clitoris. So he did. He did a clitoridectomy, and she said, "Well, now I still have the clitoral pain." So the interpretation, well, it's all in your head. Not necessarily. So a doctor called me and said his wife has this PGAD condition, and she also has a Tarlov cyst. I never heard of a Tarlov cyst. I looked it up, and there's cysts that typically form in the spine on sensory nerve roots, and Isidore Tarlov described that. He was a surgeon. Described that, and it's a fluid-filled blister. It's filled with cerebrospinal fluid that forms a blister on the nerve roots. But apparently, the typical region is on the sacral nerve roots of the location of the Tarlov cyst. And I said, this is really intriguing because the dermatomes, the sacral 2 dermatome is for the clitoris, and the sacral 3 dermatome is for the vagina. That's the sensory input. With Erwin, we started looking at in these cases, looking at actually, I started at Rutgers. I asked women to send me in a support group, PGAD support group, send me their MRIs. And one after the other, and I consulted with our chief of neuroradiology, Wei-Jen Lee, and he said he had studied, he looked for Tarlov cysts. He never saw so many Tarlov cysts in his life. We got a typical percentage is about up to 9% of people who complain of lower back problems come in with Tarlov cysts. But we found in 25 women who sent the in 25 women who sent us their MRIs, 18 of them had Tarlov cysts. That's 3/4 of the people. Sorry, it was 24. It was 18 out of 24. 3/4. We started studying Tarlov cysts in relation to PGAD, and we find here is Tarlov cysts. These are two T2 weighted MRIs, not functional MRIs. Right at S1, S2 here, this is two Tarlov cysts in this particular case. Here is a Tarlov cyst at S2 near S3. This is a Tarlov cyst. We collaborate with Chol Kim, who's a surgeon, spine surgeon, who does this surgery. This is the Tarlov cyst. It's fluid filled. You can see some aberrant nerve fibers. Here's the S2 nerve root. We find in men, this was our first case. I did it in collaboration with Erwin. This was a man who had terrible pain as he approached orgasm. It turned out that he had 4 large Tarlov cysts, and this is what got Erwin really interested in this whole thing. He had surgery to treat the cysts, and after 13 months, it took, right after the surgery, this is a major surgery, it was gradually improving, and after 13 months, he said his sex life is normal. He has a girlfriend, and he said he only had one orgasm in his entire life before he had it treated with surgically treated. We also see in cases that didn't have Tarlov cysts, we see that there's a herniated intervertebral discs in many of these cases. A herniated, here's a disc, this is abnormal. It's a bulge in the disc. First, as a diagnostic procedure, Chol Kim injects local anesthetic, sees if the symptoms are alleviated, then he does the minimally invasive surgery to reduce the bulge, and this effectively treats the condition. This particular person had he was suicidal. He was a young male. He said without doing anything, he had 8 orgasms a day. That was his average. 8 ejaculations a day. He couldn't go to work. It was too embarrassing. People are really troubled by this condition. It's very strange. So what happens in the case of these herniated discs? In all cases, it's a break in the ring around the disc. The disc has so it's sort of like a jelly donut, where the pulp is gel, like a gel, and it's a shock absorber. This is the intervertebral disc, and when this is broken, when there's a leak from traumatic injury, a fall, or even a car accident, things like that, the pulposus, it leaks out, and it's a foreign protein that irritates the nerves. But we also see cases of delayed ejaculation from a bulging disc, or no ejaculation. And when we asked the men, did it start out this way? They say no. Typically, it started out years ago that they had a lot of genital pain. So this suggests a syndrome. Well, here is my schematic representation of the three critical nerves: the pelvic nerve, the pudendal nerve that carries a sensation from the external genitals, the skin, the scrotum, the perineum in males, and the vestibule, the labia, the perineum in women, and the clitoris, of course. And then the pelvic nerve, that's the visceral sensory nerve, it's as well as parasympathetic, carries sensation from the internal organs, probably the clitoris as well, the corpora cavernosa, then the vagina, the cervix, rectum, and same the comparable homologous structures in men, and it leads. So the herniated disc is here. So if this is irritating the nerve pathways, the Tarlov cysts or the herniated disc, it can produce the symptoms that are remote. So the cause of the PGAD or these problems that we're describing could be remote from the actual source. There's nothing wrong with the end organ with careful examination. 1 minute. Okay, I'm essentially done. So this can account for a lot of genital symptoms, and this is my last slide. That we call it a natural history of PGAD. That it can start out with there's a hypothetical formulation that it starts out as a weak just awareness, and then maybe continuous awareness, and more intense it becomes painful. That can continue unabated. It can fluctuate over time, or if it's intense, it can actually produce nerve damage, excitotoxic damage. Incessant irritation of nerves can produce, can damage them, and that could be a sequel, because in many of the cases of numbness or anorgasmia, the people say that it started out maybe years ago as pain or irritation. So, and then the positive aspect of that is that when Chol Kim resolves the problem, say of doing the disc surgery, in many cases we see recovery even after years of lack of function. That people describe recovery of function. So that's a very positive possibility. So these are my colleagues, my acknowledgments, and thank you very much for your attention, and any questions?